cell the unit of life? The, the first question is, what is cell? The cell is the basic structural and functional unit of life. Structural matlab more than 200 different type of cell in humans. And functional matlab all metabolic reaction occur in cells. Okay. Next is, cell is the smallest thing that can survive independently. Anything less than a cell cannot live independently. Second point. Third is, cellular organization is defining feature of living organization. Okay. Organism, unicellular, multicellular, two types. Unicellular matlab Monera and Protista. Multicellular higher plants and animals. Now talk about unicellular organism scientist. Okay. Survive independently and perform all its important function on its own. And first is scientist named Robert Hooke, who discovered the cell, the date cord cell. Okay, this is cellula. Now the second scientist is Robert Brown, 1831, discovered nucleus. Now the third scientist Anton von Leeuwenhoek see live cell in for the first time in 1674, and in 1950 to 60, due to the advancement of electron microscopy, many details of the cell structure are described. Now talk about cell theory. Okay. In 1838, Schleiden, the German botanist, plant are composed of different cells that makes different tissue. Schleiden এই মতবাদ অর্থাৎ এই দিস প্রপার্টিটা দিয়েছিল। In 1 year later, 1839, Swan, the British zoologist, he worked on animal cell and plant cell. The animal cell properties, the outermost boundary was cell membrane, and cell wall was present above the cell membrane. Okay, and he can conclude a hypothesis. This hypothesis is, all plants and animals are formed of cells and product of cells. And all plants and animals are formed of cells and product of cells. Now talk about cell theory was together given by Schleiden and Swan. First cell theory. All living organisms are formed up of cell and product of cell. Second is, cell formed from pre-existing cell. This is given by Rudolf Virchow. Final step of cell theory, Rudolf Virchow, 1855. It is omnis cellula e cellula. Okay, मतलब cell divide and new cell are formed of pre-existing cell. 1855. The smallest organelle is ribosome. Now overview and cell of a cell. Okay, this is the cell membrane, plasma membrane, plasma lemma. This is cytoplasm, the main arena of metabolic reaction. It keeps the cell in living state, semi-fluid matrix. This is ribosome. This is Golgi complex, membrane bound organelle. This is dense nucleus, brain of cell, contains chromosome, contains DNA. This is a genetic material. This is a endoplasmic reticulum. This is smooth endoplasmic ACR. This is a rough endoplasmic reticulum. Okay. Now talk about difference between plant cell and animal cell. Okay. The tricks is 4C. You must remember 4C. The cell wall present in plant cell, absent in animal cell. Chloroplast present in plant cell, absent in animal cell. Central vacuole present in plant cell, absent in animal cell. And centriole and centrosome absent, and here cell division present. Centrosome help in cell division. Centriole means membrane. Centriole is a membraneless. Okay. In plant cell, vacuole is much greater than nucleus. Animal cell nucleus is the middle point. Now talk about the difference between prokaryotic and eukaryotic. The pro मतलब primitive and eukaryotic मतलब nucleus. और यू मतलब true karyotic, true nucleus, primitive nucleus and true nucleus. The first difference is nuclear envelope or nuclear membrane are absent in prokaryotic, present in eukaryotic. Membrane bound organelles are absent in prokaryotic and present in eukaryotic. 70 ribosome membraneless universal protein factory. Here 80 ribosome and 17 in mitochondria and chloroplast. Monomerase is the example of prokaryotic. Eukaryotic example is etc. অনেক মনেরা বাদ দিয়ে etc. all of this is are eukaryotic. Now talk about prokaryotic cell. Okay, this is the cell wall. This blue line is cell wall. Second is cell membrane, which is flexible. Cell wall is a rigid. Okay, cell membrane is selectively permeable. And here the three mesosomes are vesicle, tubercle and lamellae. Mesosome is helps in respiration, cell wall formation, secretion, DNA replication and distribution. This is protein. Sorry, this is nucleate. This green part is nucleate, tenophore, main genetic material, single chromosome, and this red is protein. Here this is replicate and the double standard DNA present in extra chromosomally replicate independently and provide the serval phenotypic character like antibiotic resistant. This is a main important point. Here some particle is present. This is phosphate granule, glycogen granule and pigment. This is cyanogen granule. With the inclusion body membraneless, gas vacuole, Bruggenangi, purple circle bacteria, etc. And this is a flagella. In motile case, extension of cell membrane, prokaryotic flagellin protein and eukaryotic tubulin protein. This is microtubule. Okay, and this is a chromatophore, bias pigment, photosynthesis, help in photosynthesis. And this is 70S ribosome. Now talk about prokaryotic cell and basis. Includes all mononeurons, size is decreased, division much faster. This is an example. Now talk about cell envelope and its modification. Okay. In majority of the bacteria is formed by three layers. First is glycocalyx, second is cell wall and third is cell membrane. The innermost layer is cell membrane, the middle wall is cell wall is rigid and shape and structure. Glycocalyx is a carbohydrate coat outermost to protect the bacteria from the host immune system. Okay, मतलब बैक्टीरिया से होस्ट इम्यून सिस्टम हमको हेल्प करता है, मतलब प्रोटेक्ट करता है. एंड जो सेल वाला है रिजिड शेप एंड स्ट्रक्चर प्रिवेंट द बैक्टीरिया फ्रॉम द ब्रास्टिंग एंड कोलैप्सिंग. मतलब क्या बोलता है, मैंने कोलैप्स माने घर्षण नहीं करना चाहिए. सेल मेम्ब्रेन इज द इनरमोस्ट लेयर एंड सिलेक्टेडली परमेबल, इंटरेक्ट्स विथ outside world and it works together as a single protective layer unit. Now talk about cell envelope. Okay, cell envelope is a two types, loose layer and top capsule. This is a preventing H2 and ion loss in bacteria. This is a protection onto bacteria. Okay, based on the cell envelope nature, bacteria can be two types according to Gram-Christian. First is gram positive, takes and retain the strain and do not retain the strain. Okay. Now talk about surface structure. First is flagella, provide motility. This is filament longest. This is a hook structure. This is a basal body. Second is fimbriae, small bristle-like structure that provide the encouragement of bacteria to rock host tissue. Not a motility structure. This is a motility structure only. Example of motility. Flagella is a example. Fimbriae is a grip. Now talk about pili. That's a prilline protein. Tubular, tubular structure called sex pili or conjugation tube not help in mobility. Now talk about ribosome and inclusion body. Okay. Then we start the NCERT line by line reading and most important example. Okay. All ribosome all controlled in this one page. Ribosome, the universal organelle present in all cells. This is a protein factory makes protein in a translation. Pallet particle in 1953 proposed or seen the ribosome. The structure is rRNA plus protein. There is 70S ribosome can break to two odd number of a 50S and 30S. 50, 30, 70. Okay, and 80 is the middle of to add up 60 plus 40, not 100, 80. This is even at, this is odd at. E the Sedberg unit, sedimentous coefficient, etc. And if you see the this is a mRNA strand and this is a ribosome, this structure is called polysome or polyribosome. If this is a 40S and this is a 60S and now this is control as add together in the deep, then the additive is 80S. Now talk about NCERT. Okay. Line by line. Cell, the unit of life. What is cell? The first question is, what is the makes an organism living? The answer, or what is the in that to this the presence of the basic unit of the cell life. Cell is the all living organism. All livings, the all organisms are composed of cell. The main important line. Some are composed of single cell are called unicellular and some like us are composed of many cells. This is multicellular. What is a cell? The smallest unit. The first unicellular organisms are the capable in independent existence and the performing the essential function of life. Okay. Anything less than complete structure that does not ensure that hence cell is the fundamental structure and functional unit of the all living organism. Anton von Leeuwenhoek first saw and described the life cell and Robert Brown later discovered the nucleus. And the invention of microscope and the important leading of the electron microscope revealed in 1950s century. In 1950, yeah. Now talk about cell theory. In 1838, Matthias Leiden, a German botanist, explained a large number of plants and observed that all plants are composed of the different kinds of cells which form the tissue of the plants. And at the same time, they are Theodore Swan, 1839, the British zoologist, studied the different animals and in thin outer layer today known as the plasma membrane. He also concluded that based on the studies on the plant tissues that is the presence of the cell wall, the unique character of the plant's cells. On the basis of the Swan proposed, the hypothesis is that bodies of animals and plants are composed of cell and products of cell. This is first cell theory. Yes. And further, Sladen and Swan together formulated the cell theory. This theory, however, did not explain how new cells are formed. And then the new scientist named Rudolf Virchow first explained 1855 Cell divide and new cells are formed pre-existing cell অর্থাৎ omnis celluli cellula. He modified the hypothesis of the skeleton as one and given a new cell theory final shape. The cell theory is understood today. All living organisms are composed of cell and product of cell. Now all cells are arised from pre-existing of cell. Okay. Today's lecture ends here. Thank you.